Have you ever thought about those medical mysteries that seem impossible to solve? The ones hidden deep inside our own DNA? Well, today we're going to dive into a powerful genetic tool that's basically a master detective for our bodies. It's called exome sequencing. So, let's get into it. For so many patients with rare diseases, the path to a diagnosis is this long, incredibly frustrating journey. It's often called a diagnostic odyssey. They bounce from specialist to specialist, sometimes for years, just looking for an answer that feels like it's nowhere to be found. So what do you do when the clues aren't on the outside? That's the real challenge, right? How in the world do you find a single typo in the 3 billion letter instruction manual that makes up our DNA? It's a true detective story, just on a microscopic scale. And here's where you really start to see the scale of that challenge. For just one patient, sequencing their DNA can turn up this massive list of over 140,000 genetic variations. These are all our initial clues. The whole process is basically a funnel, designed to filter that huge number of clues all the way down to just a handful of the most likely culprits. It's a huge investigative puzzle. Okay, so to solve this kind of puzzle, we have to know where to look. And that really means understanding the difference between our entire genetic library and, well, the most important books inside it. So think of your whole genome as a massive library. It contains every single piece of information written about you. Reading the whole thing, it's slow, it's expensive. But the exome, that's the shortcut. It's like walking into that library and pulling out only the essential recipe books, the parts that actually tell your body how to build everything.
And this chart just drives the point home, doesn't it? Look at that tiny little sliver. The exome, all those crucial recipe books, makes up only about 2% of our entire genome. It's an incredibly targeted approach.
So why would we focus on just that tiny 2%? Well, here's the kicker. Even though it's so small, the exome contains roughly 85% of all known disease-causing mutations. We're basically ignoring all the fluff and going straight to the most important chapters in the book. All right, so we know what we're looking for and we know where to find it. But how do we actually get from a biological sample, like blood, to the digital data that our detectives can start to analyze? This whole workflow is laid out for us right here. You can see how it starts with the patient, moves through the whole lab process, like extracting the DNA and sequencing it, and it ends with that ranked list of candidate mutations. It's a really systematic journey from a sample to an actual insight. So let's break down those key lab steps. First, you gotta collect the evidence. That's the DNA extraction. Then, the DNA gets prepped and, this is the crucial part, it's enriched. This is where scientists use these tiny molecular hooks to grab only those exome recipe books we talked about. After that, a sequencing machine reads the code, turning it all into raw data for the real analysis to begin. And this is where the investigation really starts to heat up. We have all the data, but now comes the hard part, sifting through all of it to find that one variant, that one typo that cracks the case. So we're back to this massive number. The process gives us around 140,000 potential suspects. How do you possibly narrow that down to find the one that actually matters? Well, we filter them, just like a detective eliminates suspects. First, you get rid of the common variations that lots of healthy people have, because a disease-causing typo is usually pretty rare. Next, you use computer models to predict which of these variants are most likely to actually break something, like a protein. And finally, you look for variants that follow the pattern of the disease through the family tree. And after running through all those filters, that massive list of 140,000 suspects gets whittled all the way down to this. We're left with just 1 to 3 top candidates, the potential culprits. And here it is, the smoking gun. In a real study of a family with a high risk of cancer, the entire filtering process pointed to a single variant. You can see all the evidence here. The genetic code, the inheritance pattern, it all identified one specific typo in the CPXM1 gene as the cause. Case solved. Finding an answer like that can end that long diagnostic odyssey for a family, which is just incredible. But the applications for this technology go way beyond solving these rare disease mysteries. That same detective method has become a total game changer in cancer care. By sequencing a tumor's exome, doctors can figure out the specific mutations that are driving the cancer. And knowing the tumor's genetic profile lets them create a personalized treatment plan, choosing drugs designed to attack that cancer's unique weaknesses. And it gets even more personal than that. This technology is fueling this incredible field called pharmacogenomics. It's a fancy way of saying that we can use your unique genetic code to predict exactly how you'll respond to certain medications. It helps doctors pick the right drug at the right dose just for you. And all of this brings us to a really powerful final thought. The question is no longer just, can we read our genetic code? As we get better and better at this, the bigger question becomes, what are we going to choose to do with all of that knowledge?